Well, welcome to our first Safer World by Design webinar for the year. Today's topic is on the important subject of cholestasis, which is one of the most serious and common issues with regards to liver toxicology. And our speaker today, Pamela El-Azi, will discuss the prediction of the potential for this kind of cholestasis based on using HepaRG. So with that, I pass you over to our speaker. Pamela, please go ahead.
Hello. Thank you, Mr. Hardy, for this introduction. So, as said, I'm going to talk about the ability of HepaRG cell line to predict the cholestatic liability of drugs and the mechanisms involved in drug-induced cholestasis. Many hepatic lesions can be induced by drugs, including hepatitis and steatosis, but the most common manifestation of adverse drug reactions is cholestasis. Cholestasis can be defined as any situation of impaired bile secretion resulting in bile acid accumulation, either in the liver or in the systemic circulation. The clinical features of cholestasis include pruritus, jaundice, fatigue, nausea, and or sometimes increased blood levels of some liver enzymes such as transaminase or bilirubin. Depending on the site of bile acid accumulation, cholestasis can be classified as intrahepatic or extrahepatic. The synthesis of bile acid from cholesterol is a complex pathway involving several enzymes. Most of them are cytochromes P450, such as CYP7A1, CYP8B1, and CYP27A1, to produce primary bile acids, cholic and chenodeoxycholic acids. The secondary bile acids, deoxycholic acid and lithocholic acid, result from deamidation or hydroxylation of primary bile acids. Both primary and secondary bile acids undergo conjugation with either taurine or glycine. Bile acids synthesized in hepatocytes, as well as the other bile components, are excreted in the bile canaliculi, stored in the gallbladder, and discharged into the duodenum. 95% of bile acids are reabsorbed in the small and large intestine and return to the liver. That is what we call enterohepatic cycle. On the sinusoidal domain of hepatocyte membranes are localized influx transporters, whose activity depends or not on sodium. NTCP is the major bile acid influx transporter. Efflux of bile acids occurs mainly via the bile canaliculi and is mediated by transporters of the ABC family. BSEP is a major canalicular bile acid efflux transporter. It is also involved in the transport of drugs and interaction with certain drugs. MRP2 is another canalicular transporter, primarily responsible for transporting bivalent bile acids. Other bile acid efflux transporters, such as MRP3 and MRP4, are also present on the basolateral domain of hepatocytes, but they are weakly expressed under normal physiological conditions. They allow the evacuation of bile acids into the bloodstream. When the canalicular excretion of bile is altered, bile acids accumulate in the hepatocytes, resulting in intrahepatic cholestasis. The situation can have a direct or indirect impact on the expression and activity of other bile acid transporters, such as a decrease in bile acid influx, an inhibition of bile acid synthesis, as well as an upregulation of basolateral transport. Different models have been developed to assess the cholestatic liability of drugs and other compounds. The majority of current hepatic in vivo and in vitro models for cholestasis detection have shown clear limitations. Indeed, many drugs can cause cholestasis in humans without any significant liver injury in animal models. This can be due to differences in bile acid synthesis, composition, and regulation that exist between humans and animals. The most commonly used animal models to study cholestasis include bile duct ligation. Bile duct ligation is the most widely used model, but this model reproduces acute obstructive biliary lesions, which is rarely seen in humans. DDC feeding and anide feeding in mice are other useful models. In fact, DDC feeding is used to study chronic cholestasis. MDR3 knockout mice is used to mimic MDR3 defects in humans. And BSEP knockout mice is another model, but this model cannot develop severe cholestasis unless mice are treated with cholic acid. Assays using membrane vesicles cannot predict cholestatic risk of drugs that are weak BSEP inhibitors, and in addition, they lack the essential cellular components. The use of primary human hepatocytes, which is the gold standard model, is limited by the lack of availability, the interdonor variability, and the rapid loss of hepatic functions. The primary human hepatocyte spheroid model would be valuable, yet its predictive capacity of cholestatic drugs has not been fully assessed. Alternatively, human hepatic cell lines have been proposed for hepatotoxicity studies. HepaRG2 cells commonly used are of limited suitability for drug-induced cholestasis studies. Indeed, they do not express NTCP. They synthesize some bile acids, but at low levels, and this cell line is defective in oxidation of the side chain, conjugation, and transport of bile acids. They also lack or express at very low levels the major metabolism enzymes. HepaRG cell line is a human metabolically competent model that expresses features of mature hepatocytes, such as expression of phase 1 and 2 enzymes, transporters, nuclear receptors, and at levels comparable to those in primary human hepatocytes. The improved culture maintenance and stability of these cells make from them a suitable model for long-term hepatotoxicity studies. HepaRG model is a bipotent cell line. Once differentiated, you can distinguish hepatocyte-like cells that are surrounded by primitive biliary cells. In the next slides, I will talk about the suitability of the HepaRG cells as a model to predict drug-induced cholestasis and the underlying mechanisms. Immunolabeling shows that HepaRG cells express major bile acid metabolizing enzymes, CYP7A1, CYP8B1, 27A1, and SALT2A1. Interestingly, these enzymes are exclusively expressed in the hepatocyte-like cells. Endogenous bile acid content was measured in HepaRG cells and in primary human hepatocytes. Interestingly, HepaRG cells synthesized bile acids at a rate comparable to that of primary human hepatocytes. We aim to compare bile canaliculi structures in HepaRG cells and primary human hepatocytes. Phase contrast images show that bile canaliculi of HepaRG cells are similar to those observed in sandwich and the classical cultured human hepatocytes. This was confirmed by phalloidine labeling, which showed an abundant pericanalicular cytoskeletal effect in all three models. Under electron microscopy, bile canaliculi of HepaRG cells appear to be surrounded by tight junctions. Next, to compare the mean surface between the three models, we labeled the bile canaliculi using CDFDA. Bile canaliculi surface was comparable between HepaRG cells and classical cultured human hepatocytes, but was larger in sandwich cultured human hepatocytes. And then we designed a study to assess the localization of main hepatobiliary transporters by immunofluorescence labeling in both HepaRG cells and human hepatocytes. NTCP was localized at the sinusoidal membrane domain in HepaRG cells and the human hepatocytes, either in sandwich culture or classical culture. MRP3 was exclusively localized to the basolateral membrane domain in all liver cell models, with a more intense labeling in HepaRG cells. BSEP, MRP2, MDR1, and MDR3 were localized to the canalicular membrane domain in all three models. While immunolabeling of BSEP is weaker in HepaRG cells, MRP2 and MDR1 show more intense labeling in HepaRG cells compared to human hepatocytes. Importantly, transporter immunolabeling was restricted to hepatocytes and HepaRG cells, as no labeling was visualized in biliary cells. After the validation of transporter localization, we assessed their activities. Activity of NTCP was measured using radiolabeled uracolic acid. It showed that NTCP is functional in all three models, although its activity in HepaRG cells is weaker than human hepatocytes, which is in agreement with the faint labeling of NTCP observed in HepaRG cells. We also assess the activity of MRP2 using CD/FDA, a substrate of MRP2, in the absence or presence of an inhibitor of MRP2. Fluorescent CDF was visualized in bile canaliculi of HepaRG cells and the human hepatocytes. In the presence of the inhibitor, CDF was not effluxed to the bile canaliculi. Activity of MDR transporters was also assessed using GC1 dye in the absence or presence of verapamil, a MDR inhibitor. Cells that are labeled in green correspond to hepatocytes exhibiting important MDR activities. In the presence of MDR inhibitor, GC1 is accumulated within the hepatocytes, and red fluorescence is observed in both cell types, the hepatocyte and the biliary-like cells. Activity of BSEP was measured through canalicular efflux of radiolabeled taurocholic acid, which is primarily a BSEP substrate. Canalicular efflux, basolateral efflux, as well as diffusion were measured in cell lysates and buffers and were found to be relatively similar in all three models. Time-lapse microscopy, as we are seeing in this video, allowed to reveal spontaneous rhythmic motility of bile canaliculi of HepaRG cells. This motility is characterized by repeated opening and closing spikes every 20 to 30 minutes. These spikes allow dynamic evacuation of canalicular content from what we can call saccular bile canaliculi into tubular bile canaliculi. Therefore, these spikes are associated with contraction and relaxation of saccular bile canaliculi. Comparison with classical cultured human hepatocytes showed similar rhythmic movements, as we can see here. In sandwich cultured human hepatocyte, although it is possible to see these contraction relaxation cycles, spikes are not detectable. The spike rhythms were analyzed in the presence of two cholestatic drugs, chlorpromazine and fasudil. As we can see in these videos, chlorpromazine induced permanent constriction of the saccular bile canaliculi, whereas fasudil induced strong dilation, noting that both chlorpromazine and fasudil led to a total disappearance of spikes and a loss of connections between the saccular and the tubular bile canaliculi. Bile canaliculi were further assessed in the presence of different compounds, while some, such as troglitazone, induced bile canaliculi constriction, others, like bosentan, induced bile canaliculi dilation. Importantly, cyclosporin induced bile canaliculi dilation at low concentration and constriction at high concentration. It was possible to quantify these canalicular alterations by measuring the canalicular surface. As we can remark, drug-induced constriction appeared to be irreversible, whereas dilations can be transient, as seen with the tolcapone. Non-cholestatic compounds have no effect on bile canaliculi surface. Using HepaRG cells, a study has shown the involvement of Rho kinase and myosin light chain kinase signaling pathway in bile canaliculi dynamics. Briefly, phosphorylation of MLC2 allows contraction of bile canaliculi, whereas dephosphorylation leads to relaxation. So Sharanek et al. Demonstrated that untreated cells showed alternating regular phases of phosphorylation and dephosphorylation of myosin light chain 2 every 30 to 45 minutes, indicating a role of myosin 2 in controlling bile canaliculi dynamics. Treatment with chlorpromazine induced permanent myosin light chain phosphorylation, which in fact explains the permanent constriction of saccular bile canaliculi in chlorpromazine-exposed cells. By contrast, fasudil permanently inhibited myosin light chain 2 phosphorylation, which explains the strong dilation of bile canaliculi in fasudil-exposed cells. On the basis of this study, the effect of a large set of cholestatic and non-cholestatic drugs on the phosphorylation of myosin light chain 2 was analyzed. Western blot analysis of MLC2 showed an increased phosphorylation with drugs known to induce bile canaliculi constriction and a dephosphorylation tendency with dilatators. All the non-cholestatic drugs did not significantly affect MLC2 phosphorylation dephosphorylation cycles. Here I will take chlorpromazine as an example of cholestatic drugs. We investigated the underlying mechanisms of chlorpromazine-induced cholestasis using HepaRG cells. Generation of oxidative stress was detected in hepatocytes as early as 15 minutes after chlorpromazine treatment, as shown by DHE staining, and was totally prevented up to 6 hours by the antioxidant NAC co-exposure. Moreover, expression of the 3 oxidative stress-related genes was induced in chlorpromazine-treated HepaRG cells. Rust generation was associated with altered mitochondrial membrane potential, as evidenced by the green color of GC1 within the hepatocytes. This impairment was prevented by NAC co-exposure. Pericanalicular distribution of F-actin was disrupted, and constriction of bile canaliculi was evidenced as early as 30 minutes. Next, we assessed the efflux of taurocholic acid following 30 minutes exposure to chlorpromazine. A decrease in canalicular efflux of taurocholic acid was observed and resulted in an intracellular accumulation of this bile acid, whereas basolateral efflux remained unchanged. Taurocholic acid was then assessed at different time points of chlorpromazine exposure. Inhibition of efflux was not observed before 30 minutes, whereas maximum inhibition occurred after 2 hours. Efflux activity was restored by co-exposure to NAC. All these observations suggest that oxidative stress precedes canalicular efflux inhibition in chlorpromazine HepaRG treated cells. In addition, NTCP activity assessed by measurement of taurocholic acid uptake was inhibited starting after 24-hour exposure to chlorpromazine. Importantly, this inhibition was not prevented by the antioxidant co-exposure. Then we did a transcriptomic study to assess the effect of chlorpromazine on the mRNA expression of main bile acid transporters and metabolizing enzymes. Chlorpromazine inhibited the expression of NTCP and BSEP in agreement with the activity decrease. It also inhibited MDR3 and CYP8B1 and induced the expression of MRP4. In fact, the downregulation of NTCP expression that is involved in bile acid efflux and CYP8B1 that is involved in bile acid and synthesis, together with the upregulation of MRP4 expression, which is involved in basolateral efflux, could be considered as compensatory mechanisms and protective response against chlorpromazine-induced cholestasis. Then we compared this cholestasis to cholestasis induced by bile acids overload. So HepaRG cells were incubated with 2 primary bile acids, cholic and chenodeoxycholic acids. Cytotoxicity was only observed at high levels, high concentration of bile acids. Rust generation was detected in hepatocyte-like cells starting 6 hours of exposure to bile acids. Importantly, no alteration of the mitochondrial membrane potential was observed before 6-hour exposure to bile acids. In addition, we analyzed gene expression. The changes were almost similar between chlorpromazine exposure and high concentration of bile acid overload. Indeed, at high concentration, bile acids downregulated BSEP and MDR3. They upregulated oxidative stress-related genes and downregulated NTCP and CYP8B1. Cholic acid, but not chenodeoxycholic acid, induced MRP4 expression. Another study showed that glino-chenodeoxycholate, but not taurocholic acid, induced an increase of lactate dehydrogenase release in HepaRG cells, as well as in human hepatocytes. Importantly, HepaRG cells showed a toxicity profile more similar to primary human hepatocytes than HepG2 cells that showed much more release of LDH at lower concentration of GCDC. Noticeably, when toxicity was assessed by propedium iodide, uptake by propedium iodide in HepaRG cells, GCDC appeared to be cytotoxic to hepatocytes only and not to primitive biliary cells, which lack bile acid uptake transporters. Moreover, caspase-3 activity was measured in GCDC-treated HepaRG cells in the absence of presence of caspase inhibitor. GCDC didn't trigger caspase-3 activation. Again, HepaRG cells model the response of primary human hepatocytes to GCDC, previously shown to die by necrosis and not apoptosis following GCDC exposure. To study the accumulation of toxic bile acids in the liver in drug-induced cholestasis, a study using a large set of cholestatic and non-cholestatic drugs investigated the accumulation of bile acids in HepaRG cells after 24-hour exposure to a mixture of exogenous bile acids. As we can see, only cholestatic drugs cause cellular accumulation of bile acids, while non-cholestatic drugs showed comparable levels of bile acid accumulation to those in control cells. Importantly, this cellular accumulation was characterized with more unconjugated than conjugated bile acids. So, cellular accumulation of 3 hydrophobic bile acid, deoxycholic acid, chenodeoxycholic acid, and lithocholic acid in their unconjugated and unsulfated forms was observed with all cholestatic drugs, noting that intrahepatic accumulation of hydrophobic bile acids is considered as the hallmark of cholestasis. Importantly, while LCA was not detected in control and non-cholestatic drugs group, it became accumulated in its unconjugated form after exposure to cholestatic drugs. Hence, LCA can serve as a robust biomarker to flag cholestatic drugs. And interestingly, serum LCA was found to be dramatically elevated in cyclosporin A-treated patients with hepatitis and in intrahepatic cholestasis of pregnancy, which supports these findings. Recent studies have shown that bile acids might induce liver cell injury in an inflammatory context, and that inflammation can be a feature of cholestasis. Indeed, expression of genes involved in inflammatory response was enhanced in HepaRG cells exposed to cholestatic drugs, both in the presence or absence of bile acids. Thus, we aim to question as to whether inflammation is an important factor in chlorpromazine-induced cholestasis or not. So we co-exposed HepaRG cells to chlorpromazine with either interleukin-6 or interleukin-1 beta for 24 hours or 5 days. Most effects of co-exposure to cytokines and chlorpromazine were amplified or only observed after 5 daily exposures. These effects included increased cytotoxicity, overexpression of oxidative stress-related genes, and decreased anti-CP mRNA levels and activity. An endoplasmic reticulum stress can be a feature of drug-induced cholestasis. We demonstrated using cyclosporin A, known to cause dose-dependent cholestasis, that in addition to oxidative stress, ER stress is a major cause of cholestatic effects of this drug. Indeed, 4 ER stress-related genes were strongly upregulated after 6-hour exposure to high concentration of cyclosporin A. The induction of ER stress was also analyzed after antibiotic exposure using thioflavin T dye. An early onset of misfolded protein accumulation was revealed by the enhanced fluorescence of thioflavin T in HepaRG cells and primary human hepatocytes after exposure to cholestatic antibiotics, but not non-hepatic antibiotics. ER stress sensors of the unfolded protein response were then investigated. Western blot analysis showed 2 to 3-fold enhanced phosphorylation of these 2 ER stress markers. In agreement with these results, mRNA expression of ATF4 and CHOP that are downstream of EIF2 alpha were upregulated by these antibiotics. So far, all the shown results were obtained using 2D monolayer HepaRG cells, but HepaRG cells can also be used to form spheroids. So cultivation in 3D configuration offers many advantages, such as long-term maintenance, extensive formation of cell-cell contacts, and also production of extracellular matrices. Similar to primary human hepatocytes, HepaRG cells can aggregate into compact spheroids that express BSEP and MRP2. Similar to primary human hepatocyte, expression of BSEP was inducible upon exposure to bile acids. Primary human hepatocytes and HepaRG cells were exposed to cholestatic or non-cholestatic drugs in the presence or absence of bile acids for 8 or 14 days. A synergistic toxicity of bile acid and cholestatic drugs was observed at 8 days in both primary human hepatocyte and HepaRG spheroids and was exacerbated at 14 days. No increase of toxicity was observed for non-cholestatic drugs in the presence of bile acids. Moreover, the known pattern of chlorpromazine-induced cholestasis is recapitulated in primary human hepatocytes and HepaRG spheroids. Indeed, repeated exposure to chlorpromazine resulted in the inhibition of the canalicular efflux of a derivative of taurocholic acid and its accumulation within the cytoplasm. Chlorpromazine downregulated BSEP mRNA expression in both spheroid models, reduced F-actin expression, and disrupted its integrity in both primary human hepatocytes and HepaRG spheroids. An increase in the expression of oxidative stress-related genes was noticed in both spheroid models after co-exposure to chlorpromazine and bile acids. At the end of this webinar, I'm going to conclude with the adverse outcome pathway of cholestasis set up by Dr. Vinken and his team. Indeed, all the known initiating events of cholestasis, such as alteration of bile canaliculi dynamics, cytoskeleton architecture alteration, as well as transporter activity changes, can be assessed in HepaRG cells and were found to induce bile acid accumulation. Consequently, two kinds of response are set up, and both can be developed by HepaRG cells. An adverse one consisting of oxidative stress, ER stress generation, mitochondrial impairment, and cell death, and an adaptive response or compensatory response by activating several nuclear factors to recover bile acid homeostasis. Altogether, these data highlight HepaRG cell line as an appropriate model to study drug-induced cholestasis and to investigate cholestasis adverse outcome pathway. Thank you all for joining this webinar today, and now I'll be happy to take your questions.